Hi, everybody. My name is Dana Howard, and I am a member of the Center for Bioethics and the Philosophy Department at The Ohio State University. Um, Welcome to the Center for Ethics and Human Values Monthly Care panel. This is sponsored by the Center for Ethics and Human Values, and the CARE program aims to support Ohio State's shared values in the domain of research by building a community around discussions of research integrity. The CARE program is presented with support by the OSUMC Center for Bioethics and the Clinical and Translational Science Institute. And we're so happy to have all of you here. I know this is a busy time, and I'm really excited to have this important discussion with you all. Today, we'll be discussing the ethics and feasibility of ending animal models for preclinical research. As AI and computational modeling transform research, scientists are reexamining the role of animal models in scientific research and drug discovery. And today we will explore the ethical and the practical implications of reducing animal testing in light of recent federal push to do so. We're going to be considering the possibility, the limitations, and the trade-offs of technical innovation in advancing humane and effective research. And I am so delighted to have our three panelists. First, we have Dr. Christine Petersen, who's Associate Dean for Research at the College of Veterinary Medicine at OSU. Dr. Petersen is the Robert H. Rainier Endowed Chair in Industrial Veterinary Medicine and Research. And besides her ongoing research in One Health and veterinarian research, Dr. Petersen is a member of the National Academy's Board on Animal Health Conservation and Research, formerly the ILAR, and was recently part of their workshop that included discussion of new approach methods, which we're going to be talking about today. We also have Dr. Naomi Scheinerman, who's an assistant professor of bioethics in the College of Medicine at Ohio State University, a political theorist and bioethicist specializing in democratic theory and moral and political philosophy. Dr. Scheinerman's work engages with ethical and regulatory and legal questions in science, medicine, and technology. And currently, Dr. Scheinerman is co-leading a study exploring digital twins and in silico trials in drug development. And then we also have Dr. Seth Weinberg, who's Associate Dean for Research, College of Engineering, professor at the Biomedical Engineering at OSU. Professor Weinberg leads an active research program in computational physiology and has been continuously funded by the National Institutes of Health and has been employing many of the novel approaches that we're going to be discussing today. Unfortunately, Professor Allyson Bennett will be unable to join us today. So I'm going to set the scene as best as I can with some sort of preliminary remarks about the current regulatory landscape and sort of the ethical guidance that we have. And then we're going to have our OSU panelists sort of present from a very interdisciplinary lens their perspective on the trade-offs, what is exciting, the opportunities, and also the limitations of this recent push. Then we have some preset questions that I'm going to ask, but as audience participants, you also will have plenty of time to answer questions from the audience. So if you look down at the bottom of your screen, there's a Q&A little button. Feel free to ask questions throughout the presentation. You don't have to wait until the end, and we'll have those be a component of the conversation. All right, so to no further ado, I'm going to share my screen. Let me see. Now I'm feeling like I'm being put on the spot. Can folks see my screen yet? Probably not. Let me see. Now people can see it? Great. And now, all right. So the end of animal models for preclinical research. So one thing, I've been thinking about this fairly recently because there has been what seems like a push both at the FDA and the EPA this year to move away from requiring animal models. But there is actually this sort of ongoing sort of movement or inertia to have done this in a while. So I thought it would be useful to just let everybody know sort of how things have been working. So, under the first Trump administration, the EPA said it would reduce testing in mammals by 30%. By 2025 and eliminate animal testing entirely by 2035. There was concern at the time that evaluating compounds based solely on these novel, the new approach methodologies may end up harming both humans and the environment. And there has been just sort of a forward push with the Biden administration moving from the EPA to the FDA. One of the things that they did, the administration itself removed the FDA's longstanding requirement that all drugs must be tested in animals, although the FDA did not yet issue a final rule to that effect. And then this year, both in the FDA and the EPA and the National Institutes of Health, there has been this movement to at least not require animal models when it comes to research, and also to encourage using novel approach methodologies in different sorts of ways. And this has been gaining momentum for a while. So we just sort of see, in 2000, only 1% when it comes to biomedical research, only 1% of NIH funding was related to novel approaches. And now it's up to 8% in 2024. And so, not only is this that's sort of like the push to use novel approaches. I do also want to highlight that when it comes to animal use, there is a lot of regulatory oversight to ensure the humane and ethical use of animals in research. And in particular, the Public Health Services Policy on Humane Care and Use of Laboratory Animals, which was passed in 1985, has required that all federally funded research institutions incorporate an animal care and use committee oversight board. And the responsibilities of an IACUC, this may be very familiar to many of you in the audience, is to ensure throughout the life cycle of the research project that there's the minimization of harm for animals through a number of mechanisms. And one of the ways in which the IACUC works, and for many of you, this might be also a refresher, but it's important to highlight because this is going to be in some ways a key feature of the discussion that we're going to be having today, is that a lot of the ethical underpinnings of the use of animals in research involves the 3 R's. The first one is this idea of reduction, which means reducing the number of animals used in experiments to the bare minimum that is required for the scientific validity of the study. So there's a number of ways to do that, including improving experimental techniques, improving data analysis techniques, and also sharing information and data sharing across studies so you don't have to continue doing sort of the same studies over and over. There's also the importance of refinement, which is the idea that we need to refine the experiment or the way the animals are cared for so as to reduce their suffering. And this is in some ways a lot of what the purview of the IACUC is supposed to do, which is, ensuring less invasive techniques in your study methodology, incorporating better medical care for animal subjects, and also better living conditions for animals. And then finally, there's the sort of push for replacing experiments on animals with alternative techniques, such as using cell cultures instead of whole animals, computer models, using human volunteers who can consent to research rather than animal models, and using large-scale epidemiological studies. So those are the three R's. And I mean, one of the things sort of to prime the ethical discussion that we're about to have, relying on animal models is not perfect, right? So traditional animal models often do fall short of predicting human outcomes. One study found that more than 90% of drugs that appear safe and effective in animals then fail in the translational phase in human trials due to safety and efficacy concerns. There's also potential limitations related to supply chains, including, how to ethically engage scarce non-human primates. So there is an urgency to potentially adopt human relevant alternatives. But there are also ethical trade-offs of moving away from animal models. And so hopefully we can really dig into those today. Some of the trade-offs that I kind of hinted at are just thinking through trade-offs for the general public about the sort of scientifically valid outcomes that we end up having. And there's also, I think, interesting trade-offs specifically for human research participants who are then going to participate in research. And also, I hope that we also have a chance to talk about potential trade-offs for non-human animals, who, how do we gain generalizable knowledge related to non-human animals in this sort of push to use methods that are different from animal models. So those are sort of big picture questions that I'm interested in. I think I'm going to now let Christy move on next, and you could share your own thoughts on this. Then Naomi, and then Seth, you could sort of go after Naomi. And I'm going to stop sharing my screen. Let me see if I can do this. I did it. Great.
Great, thanks, Dana, and thanks for that really useful introduction. So I'm the Associate Dean for Research at the Veterinary College. I'm also a veterinarian. Those two do not always follow, but it does in my case. And, it's funny because we talk about, animal models. As humans, we like to think that we're, the best and not animals, but we are. And one of the things I always remind people of is a lot of what we know about the anatomy, physiology, and pathology. So like all the ways that our bodies work and then the ways they go wrong, we actually have learned through using dogs and pigs and sheep and mice and other creatures, much more than we have by doing experiments on humans. Thank goodness, right? So when we talk about how things work, we've always understood it in this kind of comparative, collaborative way. So I'm someone that works on a infectious disease that dogs are the reservoir for. It does kill dogs and it also kills people. It's called leishmaniasis. It's a neglected tropical disease. And what we do is we look for ways that we can both prevent infection from the dog to people to protect people, but also, that would protect other dogs from getting infected. It also teaches us things as a model system about the immune response and how, the skin works to lead to infections. And all of that is what's considered kind of dual purpose research. So there is a lot of biomedical research that is done in creatures where it is to benefit humans as written, usually to a funding agency like NIH. But it turns out it really is very useful for the creature we're actually doing the research on. And I think that's sometimes lost in discussion because using a natural animal model of a creature that actually normally gets infected by this, is dying of it out in the world is something that people forget. And we think that, we're just doing things to creatures in cages that's completely experimental. So it's in that setting that I have lots of conversations with lots of people about, how animal research really is also veterinary research. And it does help the health of animals. So as we move forward in this conversation, I think that's going to be one of the things that I will remind and highlight. I also was a nerd who lived in an epidemiology department for more than a decade. So I'm pretty into, really robust study design and making sure that we are using the right amount of creatures to find a result if you're going to find one. And that's part of the uh, refinement of experiments to make sure that we're using the best model in the best way. And I think at the end of the day, that should be a practice we've always had, but it hasn't always because some animal models are just easier. Um, and I think then that is now the push is to make sure that, we use the system that makes the most sense to figure out what we're trying to learn. So with that, I will doff my hat back to Dana, and look forward to further discussion.
Thank you, Christy. Naomi, why don't you go next?
All right, great. I'm going to share my screen. And is it in presenter mode
Yes, it is.
for everyone? Okay, great. Thank you so much, Dana. Thank you everyone else for being here and for the introduction and including me in this panel. I'm looking forward to this conversation. So today I'm going to speak about both what Dana referenced, the project that I'm on, as well as this perspective piece that we wrote that's currently, most recently, we submitted a response to reviewers at Nature Digital Medicine entitled From Animal Models to Digital Twins, Realizing the FDA's Vision of New Approach Methodologies. All right, so the plan is to just give, by way of introduction and background, what are we doing on this project and how did it help inform our main arguments in this perspective piece. So this study, funded by Arnold Ventures, has a goal of producing actionable policy frameworks to guide the safe, equitable, and evidence-based use of digital twins in in silico trials in clinical drug development. So we're looking actually at it after animal research would have been done in the phases 2 through 4, where there's fewer actually studies being done. But as a result of sort of thinking about it, we had thoughts on like the preclinical stages. So our project team is quite excellent, coming, especially my co-leaders, Ravi Parekh and Holly Fernandez-Lynch, and I wanted to give a special thanks to Ashley Eady, our excellent postdoc. She prepared some of the slides that I'm using and also was the first author on the perspective piece. Okay, so what are digital twins? I know Seth is going to talk a bit about this, so I'll try and give like a brief overview. Digital twins are computer-based simulations of individual patients or their organs that allow researchers to study potential interventions, learn properties of diseases, and extract further information about system functioning. They input data such as from electronic health records and drug databases in order to generate predictive statistics about how a disease may progress in that individual or how a drug dose may be optimized. So some applications of this are drug and dose optimization, organ-specific modeling, end-of-one trials, such as for small patient populations, such as rare disease, interim clinical trial adaptation, and expanding clinical indications. So one example in particular from rare disease, for Huntington's disease, a digital twin allowed researchers to uncover a novel target related to cognition and motor function in the disease. And although there was recently, maybe you heard in the news, a gene therapy that shows a lot of promise in reducing about 80% of onset of symptoms in patients. That was not using this digital twin framework, but perhaps some of the digital twin information that we learned contributed to further understanding of the disease. So then what are in silico trials? So in silico trials are computer-based simulations that are intended to replicate traditional clinical trials using virtual patient cohorts to test the safety and efficacy of drugs or medical devices. So whereas a digital twin is a virtual representation of an individual organ, system, or whole patient, in silico trials are whole clinical trials of a particular population. And thus far, there have been some drugs identified, but not yet proved on the basis of using in silico trials that have later reached market after clinical trials actually took place for a variety of diseases, including HIV-1 inhibiting drugs, anti-cancer agents, such as the ones listed here, and some kinds of antibiotics. Okay, so that brings us to April 2025, as Dana introduced. A project began in January of this year, 2025, with doing some background work into a scoping review and starting to think about the qualitative interviews that we wanted to do with 4 key stakeholders: regulators, academics, industry representatives, and patient advocacy members. And when this FDA announcement came out, and subsequent roadmap to phasing out animal testing and preclinical safety studies for pharmaceuticals, we had thoughts. So this announcement largely outlines, as Dana mentioned, the agency's roadmap for implementing FDA Modernization Act 2.0, a 2022 statute that amended the Federal Food, Drug, and Cosmetic Act to remove language explicitly requiring preclinical animal testing and drug development, and authorize sponsors to use non-clinical tests, such as what are referred to as NAMs, or new approach methodologies, to investigate the safety and effectiveness of new drugs prior to testing in humans. So in so doing, the roadmap makes the case for the importance of NAMs, given that, and they cited the same statistic that Dana mentioned, 90% of animal-tested pharmaceuticals do not make it to market. The roadmap mentions in silico trials in particular, as replacements specifically, writing, overall, in silico NAMs may act as powerful adjuncts or replacements for animal studies by predicting human-relevant outcomes through data and modeling. They are rapid, cost-effective, and can integrate vast amounts of existing knowledge. For instance, an AI model might instantly compare a new drug to hundreds of prior ones to assess risk, something impossible with animal testing alone. Importantly, as regulators gain confidence in these tools, they continue through retrospective validation and prospective pilot use they mention. They could be formally adopted to reduce or replace specific animal tests. So in our capacity, as people who are thinking about the regulatory policy and ethical implications of using in silico trials, we penned a perspective piece arguing the following. To realize this trajectory towards new technology-mediated advancements in preclinical and clinical drug development, regulatory agencies must urgently address the following. First, fundamental certification standards, meaning regulatory agencies should adopt platform-based certification, allowing a single NAM or digital twin model to be validated across multiple therapeutic contexts, once it has demonstrated core performance standards, such as predictive accuracy with known human responses. Second, current resource allocation challenges. So the regulatory framework in FDA itself is not currently, we fear, equipped to evaluate these alternative methods of verifying drug safety and establishing validity, as no pre-established framework exists. Staffing at the FDA is also not up, we believe, to the challenge currently, given recent significant staff cuts. And there's substantial institutional modifications in labs across the nation that would have to transition to NAM systems in ways that may actually slow drug discovery and development, certainly in the short term. Thus, effective NAM implementation, we write, requires coordinated investment across several major areas: workforce pipeline development through strengthened academic training programs and university research initiatives, enhanced NAM training for current regulatory reviewers, and infrastructure development for international NAM-based data repositories, such as server clusters and secure sharing frameworks to facilitate model validation. So speaking of validation, scientific validation frameworks for NAMs. So now that we're a bit farther along in our study, also, we're sort of in the thick of the interviews and wrapping up the scoping review and all of the literature review that we've had to do for that. Validation is like perhaps the most important epistemic and moral value that has arisen here. As of now, the FDA just doesn't have a validation framework in place for confirming whether these technologies are predicting what they claim to predict, and thus we need to establish clear protocols for weighing different types of evidence and determining acceptable risk-benefit profiles for regulatory approval. And that's it. Thank you very much.
Thanks so much, Naomi. And Seth, why don't you go next, just sort of present your own perspective.
Yeah, great. Well, thanks again, Dana. Thanks for coordinating this, and thanks to my fellow panelists as well. As Dana mentioned, I'm the Associate Dean for Research in the College of Engineering and a professor of biomedical engineering. And my lab is broadly focused on computational physiology. Our primary focus is related to cardiac electrophysiology, so looking at the electrical signaling in the heart. And so I'll talk, quite a bit about my perspective from, looking at in silico and the development of new techniques there. But my experiences, I think, are also pretty shaped by kind of my whole training process, in particular my PhD work, where before I jumped into the computational end of things, I was a pure experimentalist doing animal work. Um, and so I can talk a little bit about that as well. I think, from my perspective, when I think about, kind of the overall philosophy of like how we design biomedical research, there's a really cliche saying that we learn in the modeling field that I think and I apologize for using it for those that have heard it, but I think it's really helpful and applicable, which is the saying that all models are wrong, but some are useful. Right. And, we often will talk about that in the context of computational models, but I think, especially in this conversation, it's really applicable to all models, right? Including thinking about animal models, right? And so, I think it's really important to think about, with whatever system we're doing our experiments and analysis on, if it's intended to represent something else, we know that it's incomplete, right? Whether that's a set of equations or whether that's using a rat to represent how, a human is going to respond to a particular drug therapy. Um, and so I often think about that in the context of, how are we deciding and defining the particular either experimental and then, computational work that we do. So my PhD work, which I'll mention here, which I think is really I think it's really illustrative in highlighting both kind of the challenges of replacing animal research, but also highlighting, I think, why there's really big opportunity for it. And so specifically my PhD work was looking at developing essentially a new technique for terminating cardiac arrhythmias, essentially kind of optimizing what defibrillators do and in theory also trying to reduce the pain that patients would experience when they get these kinds of electrical shocks. Um, and so we had this overall hypothesis of how we might potentially do that. And so the work essentially kind of went through sort of multiple stages of animal models. So starting initially at rat and then guinea pig and then pig. And in the cardiac world, that's kind of getting increasingly closer to human cardiac physiology, right? A rat is obviously very different than a human, whereas a pig is relatively close. And at the same time, each of those kind of went from like an in vitro stage to an ex vivo and to in vivo. And so with each of those stages, we learned something. We kind of refined our understanding of how this potential method actually worked. And so when we got to the next stage, we were already starting from a more optimized place. We could reduce the sample size. We also had a better understanding of what might actually work in humans. But we also found the things that worked at the lower stage didn't necessarily work as we got closer to more human-like. And in particular, we found that big jump, in particular, when we went to in vivo. And I think that, in particular, kind of it highlighted, I think, really where a lot of the big challenges are is, when we look at isolated systems often in our biomedical research, right, we can understand how that system is going to very cleanly respond. But in, a living human and in a living animal, we're not isolated systems, right? They're all highly coupled. There's lots of crosstalk. If you perturb the heart, it's going to impact almost every single other system in the body, again, whether we're talking about an animal or a human. And so, the particular thing that we were studying still ended up actually working a little bit better than we expected, but we identified a whole lot of challenges. And so I think from my perspective, I think one of the biggest challenges with ending animal work is going to be to figure out how can we get a better understanding of how the systems that we often think about in isolation actually integrate and have crosstalk in living animals, right? Again, whether we're talking about animals or humans. It's interesting that on that project, we actually also had a computational modeling collaborator. And again, this was many years ago. I won't throw a number out there. And so for technical limitations, actually, the computational work couldn't really do at scale what we could do experimentally. But now, many years later, if we were going back and doing that project, I think the modeling could have done significantly more heavy lifting. It still added to the project, but we couldn't do sort of the optimization that we did in the experiments with the model, whereas now I think we probably could have. And so I think that, that sort of at least initially opened my eyes to where computational work, can in many ways provide significant utility to reducing the type of animal work we did. So in my own lab, that actually in many ways was one of the main reasons why I had a pretty hard pivot of moving to computational work. I like the idea of doing biomedical research that could, develop techniques to reduce the reliance on animal work. And so to the question of, how close are we being able to potentially, completely eliminating animal models? This is sort of a not satisfying answer, but I think the answer is it really depends on kind of what fields we're talking about. In my own field of cardiac physiology, it's relatively sophisticated in the computational world. I mean, there's really deep roots that go back to the 1950s in terms of electrical signaling modeling. And in some ways, it predates even that, of developing these biophysics-based types of models, which is the work that my lab now does. And especially in the last decade or so, it's really kind of grown specifically to the kinds of things that Naomi just brought up of using those types of physics-based models to develop digital twins that represent the individual patient. And the idea of, doing an in silico trial either at the population study, which are the kinds of things that we often do, or developing a digital twin of an individual patient, again, kind of that N of one experiment. And so I think there's a lot of utility in, continuing to progress those types of models. I think the two big challenges that I really see in kind of the computational side is one, and again, I only touched on this already, so thanks for teeing it up really nicely, is what exactly defines a digital twin is a pretty poorly defined term. And if you ask 11 scientists, you'll get 12 answers. Um, but I think starting to think through the process of how do we create that definition and, putting definitions and then regulation around it if it's going to obviously inform, clinical practice. Um, and so I think one, defining what that definition is, what types of information is needed, and that very much may be a context-specific question. What's needed in the cardiac world may be very different in the pulmonary world, very different in the cancer world and whatnot. And then I think where really there's a lot of utility kind of building on new machine learning and AI techniques is thinking about the question that I mentioned earlier, which is how do these systems integrate? Um, we can understand these individual systems in isolation, but to really understand the kind of the crosstalk between systems, we really need a lot of data. And that's where machine learning types of techniques can really thrive. And so I see that as one of the next big, really both barriers and opportunities of where I think this field will go. And I know there's groups that are working on this challenge, and we're trying to do a little bit too as well. So guess I'll stop there, and I guess we'll open up things to questions.
Thank you to you three. That was like a really nice overview of like all the different perspectives. And Seth, you sort of teed us up to just sort of a very preliminary general question to start the conversation. So given, this idea of, the three R's and the ways in which different models can be useful, how do you see new approach methodologies that use non-human animal models to support efforts in the three R's? What are you excited about? And Seth, you kind of already mentioned it, so I would love to hear from Christine and Naomi, but also what possible obstacles or pitfalls are you anticipating as like something that you are worried about? So maybe Christy, why don't we go with you, and then Seth and Naomi, if you want to add anything.
Sure. So I think it is a great time to be in science in some ways, because we do have this richness of data diversity and just analytical might that we haven't had for a while. I am the contrarian to those who think it can solve everything, because of exactly what Seth was saying. So the hat that I didn't mention is my PhD was specifically in kind of the immunology of infectious diseases. And I think while a digital twin can understand the organ itself fairly well and its physiology, it's the inputs that it gets from the endocrine system and the immune system, and how that impacts other parts of the body is what we really don't understand very well yet, and therefore don't have the databases to understand. And both in terms of digital twins, but also, the organs on a chip will be challenged by the ability to link all these things together. And and that's often where disease happens, is because there's been a tweak of the system, not necessarily in the organ that's having the problem, but it sets off a cascade. That doesn't mean these systems won't eventually get to the complexity and ability to do it. We're just not there yet. So that's where we can't yet fully in silico understand the workings of a whole body, whether it's a mouse, a dog, a cow, or a human. Um, so so I'm excited because we are starting the steps of being able to do this, and we have to do these steps to get to the final output, right? But I think it is naive to believe that the current systems we have really can give us the answers of the full body.
Yeah, I'll jump in just to like echo a lot of what Christy just said. One historical and other regulatory perspective to consider is that animal models became much more required in the 1960s after the thalidomide, uh, there are disability rights concerns in saying catastrophe, right? Like there's problematic terminology around how you describe thalidomide. But the experience of the US regulatory system in barring thalidomide because it lacked certain evidentiary standards, including animal testing in pregnant animals. There had been none done. And so after that, that was sort of the spark that there was a lot of regulatory frameworks and acts that and amendments that came after that in order to put in place the system we have now. And then there are also the 3 Rs, and there's a lot of refinement of the acts themselves, right? So there's been decades of development in thinking about how to use animals, also compassionately, although of course, like any use of animals for a lot of people will just be impossible to even call it compassionate use. But also just in terms of validation standards, in terms of scientific requirements, and changing both regulatory and epistemic frameworks for how we think about validated information is a very long iterative process. And yeah, just to echo what Christy was saying, we are very much at the beginning of that with these technologies. And there are a lot of steps in the process for drug approval. And so each of the steps is going to require its own kind of systems and validation. And there's this huge industry that's growing that wants to do this. And a lot of questions remain around how to uniform these standards, how to put them into the regulatory framework, how to prove anything. And so the concern is, yeah, how do we get there? How many decades is it going to be until we get there? Very much at play here, given our animal model history as well as a precedent.
Thank you. And yeah, that highlights also concerns about priority setting, right? So thinking about how much it's worth to develop these models right now, and how much money and resources we want to make sure to get them approved. Seth, before I see that you want to share one thing. People are already asking a lot of questions. Thank you, everybody. Keep on asking questions. Holly Taylor just asked a clarificatory question, so maybe you could also add this in your response. So setting aside your last comments related to definition, so we're still sort of in the nascent stages of like really understanding this. Could you step back and say more about what an in silico digital twin study is, how it's different from the animal models that you were using before? Yeah,
Yeah, that's for me, correct? Just making sure I understand. Yeah.
please.
Yeah, so in my space, right? So I'll constrain this to like cardiac electrophysiology, which is my world. A common, well, common's too strong of a word because these are still, relatively rare. But an example that's been, in the literature recently is specifically looking at what is called what you would say are the ablation sites for treating disorder, atrial fibrillation, right? And so I'll try not to go too deep in the weeds because I know we have a pretty broad audience here, but essentially atrial fibrillation, which is, I believe, the most common cardiac disease overall, um, is essentially very chaotic electrical signaling in your atria, which is your upper chamber of your heart. It in itself is actually not lethal, so many people actually live with this for decades. But one of the consequences is you have blood kind of pooling in your heart, and then eventually those clots can then get sent out to other parts of your body, and that can lead to things like heart attack and stroke. So it's very life-threatening, but it's not immediately lethal, which is in many ways what makes it so challenging. And so when these are identified in the clinic, the treatment for this disease is itself quite crude. There are some pharmacological treatments, but the most common treatment is to actually literally, I mean, again, I'm somewhat summarizing, but essentially like burning and killing off tissue in the atria, which are like the sources for generating this chaotic electrical activity. And so many times that works, but also many times it doesn't. And so patients then have to come back and repeat that process. And the main reason that it often fails is, you may have sort of killed the source of that initial chaotic electrical activity, but by burning and killing the tissue, you've essentially created new ones. And that's often, at least initial, in the clinic, it's often difficult to predict, right? And so one thing that you could imagine clinically is an important challenge is optimizing and minimizing exactly where you burn that tissue, right? You want it to terminate and then potentially not generate new ones. And so something that's been done now in a couple what I would call digital twin studies in our space was to essentially generate a as much as you can sort of replica, if you will, of that patient's heart. And so that incorporates a lot of like imaging data, so things from like an MRI or CT. So you have an accurate reconstruction of the geometry of that patient, which actually varies quite a bit between patients. And again, these don't generally happen in healthy patients. So these patients may have had previous, heart attacks, or they have just inherent scar tissue, which changes the electrical signaling in the heart. And so you incorporate all of that into this kind of physics-based model that actually simulates the propagation of electrical activity in that individual patient. Now, I will say there's a whole lot of things that are kind of assumed that you can't measure at an individual basis. And so that's kind of what I was getting into is it's hard to define exactly what a digital twin is. So there are aspects of that simulation that are not going to be specific to that patient. But it's been demonstrated, and this is what people are working on, that having an accurate reconstruction of the sort of overall structure and geometry is generally pretty good. Again, for at least this problem. For other problems, that's maybe not true. And so in silico, they can essentially mimic that ablation, that what the clinician would do, and then they can actually test in those simulations to see is that effective at terminating that chaotic electrical activity. And actually going even one step further, that you can even essentially kind of like optimize the whole process, right? So you can simulate that ablation, mimic what would happen in that patient, and maybe identify, oh, actually, I need to also ablate in this other region to prevent that new arrhythmia from starting as well. And so that's been done again in relatively small scale, but it's been shown to be pretty effective. And so that's what I would say is sort of an example of a digital twin. But again, it
Mhm.
really gets into the weeds of the definition because for other cardiovascular diseases, that may not be enough representation to understand how best to treat them. And actually, it really gets into some of the things Naomi brought up. Drug therapies tend to be much more complicated because drugs have a lot of off-target effects, or not necessarily even off-target, but they just target a lot of different things. And so accounting for all of those requires a lot more what I would call personalization, so more specific aspects of that individual patient.
Mhm.
But actually, I mean, that kind of gets into a little bit of what I was going to say about this question is having like regulatory definitions of like what is the right level of detail to say like I have confidence that using this in silico technique is going to improve clinical outcome, right? Because that's really the goal, right? And so for this one particular um, you know, clinical situation of ablation for atrial fibrillation, this seems to be a good level of detail, although I actually just saw a talk at American Heart Association meeting last week or last month, and they actually identified that there's a whole nother layer of information around fat surrounding the heart that also interacts with all of these things that could impact how well you can kind of predict how this patient's going to respond. Um, so that was actually one of the main things I was going to mention. The only other kind of dimension that I was going to add to this, because I agree with everything Christy and Naomi have said about kind of thinking about, integration across the systems. I think another really big aspect or challenging aspect from a computational standpoint is kind of the dimension of time. Because one of the critical components of these systems integrating is they occur on all time scales, right? They're occurring on, again, in my world, the beat to beat of the heart, but also on, weeks, months, years, right? And they fluctuate throughout the day. They fluctuate throughout the year, right? Like all of these kind of long time scale processes. Um, and that kind of introduces a challenge of how do we represent like all of these changes that are occurring on these much larger time scales. Again, we may know how to represent, the heart on the order of a couple beats, but what about the progression of heart disease over a decade, right? That's a much harder thing to predict. And so again, I think potentially some of these AI machine learning techniques, when we start integrating more clinical data, may have part of the solution, but I think it's still one of the biggest challenges in this space.
Thank you for the example, and thank you for those comments. And in some ways, we've been sort of talking, I mean, these are technical questions, right? So we've been talking sort of at the technical sphere. And I kind of want to take a step back now in the conversation and really sort of center the ethical considerations. I would just love to hear from each of you on your view. What do we owe non-human animals who participate in research? Do we owe them respect? Do we owe them safekeeping? Do we owe them compassion? Is it the same as what we owe human participants in research, or is it different? And I mean, in some ways, I feel like that sort of ethical question it's sort of underlying a lot of the conversations that we're having now. So maybe Naomi, you could start, and then Christy and Seth, you could go after.
Yeah, I've been thinking about this question, and I mean, I think ideally, and I'm speaking for myself at this point, but I think ideally, like it would be wonderful to get away from animal testing and into really validated, robust systems. I think what's interesting about, given it's sort of a consistent line also from the 3R framework. It also reduces the sort of hierarchical, like we've reduced primate testing. There are definitely studies that we're doing on animals that we should never do on humans. So we do have a sort of hierarchy still in place that is also problematic for a number of reasons. And one of the things that I've been thinking about when it comes to sort of thinking about AI as replacement for animal models is that there's a trade-off there potentially also in terms of like the environmental costs. So there's significant environmental resource use and potential negative consequences for the environment, which will also affect animals. And those are sort of like lesser known consequences, although some of them are slightly predictable. Then freeing all animals from laboratories seems like an immediate good that might happen if we have the right infrastructure in place to replace them and like benefit both animal and human health. But there's also like serious environmental considerations in terms of the resource intensive use that AI has and all of the training and like development that's going to go into that. So I've sort of been thinking there in terms of that ethical trade-off, which I realize doesn't perfectly answer your question, but I think we owe it to them to try is sort of how I'd answer.
Thanks, Naomi. And you're highlighting also not only what we owe to the animals that are part of the research, but there are other animals living in the environment that we may have responsibilities to. Thank you. Yeah, Christy or Seth, do you want to just chime in? What is it that how should we approach our ethical obligations to animals?
Sure. So the comments that Naomi made, kind of highlight this consideration of One Health or planetary health, right? Like everything's intertwined. We live in a giant ecosystem, and if you change one thing, it's going to have ripple effects, much like what we were talking about with the body, right? That's an ecosystem, but we all live in a larger ecosystem as well. So yeah, if we return all of the laboratory animals, which is a lot of mice and rats outside, that that is something that ecologically we know is going to give a lot of food to a lot of other creatures, and not necessarily lead to shiny, happy lives for those small rodents. Um, and and that has a burden, and it isn't necessarily going to be a good outcome. You could talk to Baltimoreans or New Yorkers about what happens when you leave a lot of garbage outside. We already have a decent amount of rodents in our ecosystems, and it doesn't necessarily need more. Um, but thinking about the greater impacts of energy use, of water use, of what is already happening to our planet, I think are all things that we have to have in this conversation. Not many people have taken the veterinary oath, but we do say that we're going to protect the public health. So consideration of these things are things that I take, and I think my profession takes seriously, because it is a trade-off. And I think these are fantastic tools, but I think we need to recognize that anytime you create a tool, it's with an input that has an impact. So, it's great to be having these conversations. I think we have to have them on a broader scale, and make sure that we are incentivizing to understand, the full spectrum instead of just pushing forward one side, which I'm a little afraid we're doing right now. So do we owe consideration, compassion, um, for these other animals? I obviously am pretty biased there. I have, dedicated my life to um, creating the least amount of harm and the most amount of health in all animals aside from humans. Um, so I believe in that pretty strongly. I think ethically, it's really fascinating to think about. There are multiple states who have had laws go forward, or at least be raised as personhood for other animals. I think that has challenges. I certainly believe that we need to consider what we are doing to animals in all instances. Um, but I think a lot of our society isn't willing to give up using animals for food yet. Um, and until we've really kind of had a discussion about that, I think we can't really get rid of animals in research, because if we're going to kill hundreds and millions of them a year to feed ourselves, why wouldn't we also use them to make sure that we're healthy and not having adverse drug effects? So there's kind of checks and balances, and I find it fascinating that we're ready to, really take a strident push to get rid of animals on one side, but we really aren't on the other. Which I just think is an interesting ethical question, right or wrong.
Seth, do you want to add
Yeah,
on
I
your
mean,
thoughts?
I'll chime in. I mean, I thought a lot about this question specifically from the respect side of the ethical question you brought up. I mean, I thought a lot about that during my PhD work, which in many ways is kind of what pushed me professionally to go into more computational work. Um, but when I was a grad student doing those experiments, I always went into it with the mindset of like, I want to get as much useful information out of this experiment, knowing that there were animals that sacrificed their lives for that, for these experiments. And so, you know, there were always, kind of experimental limitations in terms of what I could do. And, that was always just a part of the challenges. But I had if I had a really good cell culture that week, I would, spend 12, 15 hours sometimes in the lab getting as much as I could out of those cell cultures because they were only good for a short window of time. Um, it also was really, I struggled a lot of times as a student when sometimes things would fail for things beyond my control, right? I mean, again, for those that maybe have done cell culture, sometimes the cells just die, right? And, you maybe eventually figure out there was some type of contamination in the incubator, or a piece of equipment broke that hadn't broken in 10 years, and so you didn't think to check it before, those kinds of things and so those animals, like those experiments, didn't provide any useful scientific information, and that I always struggled with that. I mean, I found that very frustrating, both from just not honestly from the, oh, I'm a student, I want to graduate, but from the perspective of like, I know animals died to do this work and I'm not getting any useful information out of it. And, again, like that's unfortunately part of the scientific process. Like I could appreciate that as it was happening, but I still found it really challenging. Um, Whereas computational work, right, you have a typo in your code, things don't work well and you don't get any results, but like, you lost a week, right? And this wasn't at the scale of what Naomi's talking about, where it's, significant computational power, right? This is just, my old desktop running for a couple hours. Um, I think the respect aspect is something I think is really important. I think, for me, that went a lot into the motivation of how I wanted to do my work, knowing that, the animal work that I was doing and now the computational work, which is still informed by animal work, right? That provides useful information, right? We're able to learn something from what those experiments went to. I think it'd be, kind of adding to the broader conversation. uh, I think I think probably more care could go into making sure that that happens at a larger scale, right? Not just with the experiments that we do, but in terms of data accessibility, right? Having access to the experiments that other people are doing so that it informs everyone, right? So that the same experiment doesn't have to be done in, 40 labs across the world, right?
Mhm.
Um, I think we're getting better as a society about having, open access to data and sharing code and sharing, raw data for those kinds of things. But I still think in many fields we're still slow on the uptake of that. And I think, you know, informing the respect side of that in terms of getting as much information out of it, I think is really valuable. And again, it goes into a lot of the broader things that we're talking about.
Thank you. And so I'm going to jump into some questions from the audience that are related to some of the questions that we were thinking about presenting. So one question from Carrie Freed is, so given that some of the public are not aware of oversight that is in place when it comes to the use of animals in research, can you comment on the steps that are already in place to justify the use of animals, the species that are selected for the models, and the use of relative to available non-animal options? And sort of to piggyback on that question, I think that's an important one. I mean, one question I have, so all of what we're saying, there's a lot of sort of background ethical discussions about resource allocations, the use of AI, and the way in which it has an impact on the environment. So there's already public perceptions related to some of these issues. And I'm just wondering, like, how should we think about public perceptions about the use of animal models moving forward? One thing that is interesting, they keep on doing the Gallup study about the US population's view of animal research. And Americans are getting more and more like sort of uh, they're still sort of in general approve of the use of animals for biomedical research, but they are becoming a little bit more concerned about it, right? So how should we think about these public perceptions, and how should that inform policy and regulations moving forward? Maybe Naomi, maybe you should go first since you're like the sort of deliberative democracy person, and then we could ask Christy and Seth if you have your thoughts.
Oh, I thought we were gonna go with the other one first, so I have a second to like look up public perceptions of AI in comparison. But I didn't have time. So I would say that I was sort of like a lot of things I think. I have seen that Gallup poll that AI that public perception of animal testing has gone down. Um, I think that so the way I think about democratic, you know, theory and public participation is that it really demands sort of a good institutional place to conduct deliberation, that we need really good locations and selection criteria and facilitated dialogue to be able to have fruitful public participation. That sites of contestation, such as like protests outside labs, can be tricky places to try and figure out, although they're important, like social movements and like, free speech and like participation in the public sphere is really important for tracking public values in certain kinds of ways. But in order to like better incorporate and really think about things like trade-offs, like how is AI equipped and not equipped, and what further is needed, to think about maybe like even just the transition process. Like, look, it seems like this is a really important moral value. Our compassion for animals necessitates a move away from animal research in certain kinds of ways. How do we do that into basically AI models, where there might be a lot of skepticism and a lot of misunderstanding or proper understanding of, problems there. And so thinking about what I do with democratic deliberation is like think about good sites and locations for collecting diverse viewpoints to come together and talk with experts to come up with solutions. And I haven't applied that as strongly to this arena, but maybe that's a next step for me. But I would imagine it would require something like that, given public perception and given sort of necessity in some areas of like thinking about what do we demand from AI as well.
Christy or Seth, do you have any views about both how should we think about public perceptions and how it should inform the sort of policies and interventions? And another question that has come up from the audience is, what are the responsibilities of researchers in terms of communicating sort of the methods that you're choosing and sort of the underlying ethical decisions that are going into either using an animal model or using a new approach?
See, Seth is also equivocating, but I'll jump in and then you can go, Seth. So to answer the second part first, because you just chimed in with it, I think it is critically important that we as scientists, clinicians, and all the other hats we wear to engage in the conversation, to explain what we're doing and why. And honestly, I think we haven't done it well enough, and that's how we got to here. I had the pleasure of working really closely with the director of the state hygienic lab through the SARS-CoV-2 pandemic in the state of Iowa. And that meant that I did a lot of public communication, and that's why my hair is so gray right now. Kind of kidding. But what I found was that we nerdy people forget that not everybody's is driven by data. We really love data, but not everybody does. In fact, a lot of people really don't care to hear a lot about data. So it's so much more important to be able to depict what's going on through stories that resonate with people across the board. So I think that efforts to help scientists do that better are really important, and they're happening across the university, but also kind of across the scientific realm. And I think a lot of us have recognized that this is our challenge, and we have to do it. As much as we like to be nerds with each other, we need to be nerds with other people too. And it's the time that I spent, on weekends moonlighting during my PhD, sitting on the floor explaining the physiology of why their dog wasn't doing well to people who brought in their pets, to me as a veterinarian when I was still in practice. That remind me, like it's those conversations that really make a difference. I think this isn't a new conversation. We feel like it's kind of been much more underscored, but it is true, as Naomi made so clear, that ever since thalidomide and other challenges, we really have been trying to do this. Every IACUC group board has community members on it. Through the process of that, we are required to do a thorough literature search of what has been used and what other new approach methodologies are out there. But the big challenge is that the way science is incentivized, we don't publish negative data. So if something's been tried and it failed, it may not be in the literature. So that's why really encouraging the publication of everything is so important and needs to be repeated. Both the science and the message that we have to do this, because that's one of the things that's really going to be critical to make sure that we aren't wasting creatures' lives to prove something that somebody else already showed doesn't work. Off to Seth.
Yeah, I mean, I'll echo a lot of what you just said, Christy. I think, yeah, certainly agree that it's an important thing that we as a community need to engage in. Historically, we have not been great at communicating the value of the work that we do just in general, right? But I think especially, you know, the need of both what animal work, animal related research can provide, and then also what these other alternative approaches can too, right? And so I think, again, I think exactly as Christy said, we're really great about talking to each other. And I think, part of scientific training, right? I think about things as I talk about with my own students is learning to engage with our own community, right? And learning, to write and communicate and present. But we don't do as much about thinking about how to engage more broadly with our own communities, with, you know, the more general public. And so I think it's reasonable to think about efforts that we can incentivize. I mean, again, I don't think everyone has to become, a public advocate. I don't think that's necessarily in everyone's skill set or interest. But I think when we have, folks in our communities that want to do that, and perhaps are just maybe more natural storytellers, right? Because again, exactly as Christy said, right, it's conveying the message as a story, right? We don't you need to throw spreadsheets at folks, but we need to have them understand kind of the utility and value of the kind of work that we're doing. And so I think we should value that work. And there's lots of professional societies that often support a lot of this kind of work, and institutions I think do it to some extent. But I think recognizing that it's not just things to do on the side, but that it actually brings a lot of it's really a valuable thing for us to do to support our own community, right? One really great story that lands with the general public can really highlight the value of what what all of us do. And so I think it's certainly an important conversation, and we should continue to do, what we do and more.
So I'm going to just add a comment from one of the audience members that I think is an interesting sort of insight. Sorry, my video thing just went up for a second. So Zaki Sabri said, I mean, we've been talking in some ways about only a subset of animals, non-human animals. One of the things is invertebrate animals are not even considered animals that are regulatorily protected, right? And they have a really important component in basic science research and in innovation. And so, I mean, one thing that I'm interested to hear from you is related to the sort of the regulatory environment. What ways in which the regulations currently as they are allowing researchers to live up to their responsibilities when it comes to respect and well-being of animals? And what regulations or policies would you like to see changed? We've already talked a little bit about that, but if there was like one policy recommendation that you would have, what would you like to see sort of highlighted? Maybe we'll start with Seth and then Naomi and Christy.
Sure. So I'd probably need to think more than 45 seconds to say this is a definitive answer to that question. But at least one thing that kind of jumped in, and it's actually echoing, I think Naomi mentioned in her kind of intro, is really the validation, right? And having, well-defined processes, definitions around the validation of, all of this type of work. I think, because at the end of the day, right, that's where our trust comes from, right? I mean, and so I think, having, and this is, I think it's particularly challenging in the context of, kind of the AI machine learning aspect of where the research is going, because the techniques and, you know, the opportunities there are continually growing, right? And so, it's really hard to say, okay, we're gonna, as of Monday, December 1st, based on the technology and, what's been demonstrated, I'm going to make, policies and processes based on where the current state is. But on, January 1st, it could be different, right? And, maybe that sounds like a little bit of hyperbole, but in many ways, this space is, kind of accelerating in ways that are a little bit, are kind of unprecedented. And so I think it's particularly challenging to both develop a system of policies and processes that say, okay, here's how we're going to, here's the validation that needs to be done in order to trust that, this AI model predicts that, compound X is going to treat this condition in, a large enough percentage of the population, and we trust that it's not going to have, toxicity effects that are going to be detrimental before doing any kind of animal testing. Which AI model do we trust, right? Do we do it in a whole bunch of ones, right? Which version of that, right? What's the training data? Where did all of that come from, right? So defining, I think, that process itself of how to, what needs to make those definitions, I think is really, I think it's difficult, but I think it kind of has to be tackled before we can start to have some of this, implicit level of trust that we I think a society do have for, kind of the current pipeline, if you will, but then integrating these new techniques into it. I think that needs to be done, but I do see it as one of the biggest challenges too.
I mean, yeah, this is kind of the problem of a hierarchy that I just stated before without getting too much into. That you have these situations where like some animals have protections, and it's often around this connection to like higher cognitive versus lower cognitive capacity, where higher cognitive capacity usually means more human-like. And so we haven't really, known really what to do with invertebrates in certain ways because they lack what we would expect in cognitive functioning. Although, I mean, I think the other panelists will know better than me. Doesn't Aya Cook still review invertebrate applications for studies in a lot of places, even though they are like less regular, like less protected under the same standards, but they still go through Aya Cook like justificatory standards. So there is that. And I think, yeah, I think sort of like a public participation deliberative moment to really look at like how are we using animals, who's protected, who's not, and what are we transitioning to would be really important in that conversation.
Christy, is there something that you would like to see changed in the regulations? And there's something that you think that the regulatory and institutional policies are like really doing right to uphold researcher responsibility?
Well, I mean, I would like to emphasize that, the work that Aya Cook has done for several decades now, really has been critical in pushing investigators to think about the best way to use animals rigorously, to ensure that we are, when we do undertake, kind of like Seth was talking about, research that does lead to inherent harm or death of animals, that we are getting as much out of it as possible. We are using the least amount of creatures as possible. And, that there is oversight should there be undesired effects, and reporting, and then, feedback on how to do it better. I, like many, have been paying attention to the work done in cephalopods lately. So this is octopuses and squid, octopi and squid. And because of that, there has been a request for information on further guidance to assured institutions on cephalopod care and use. I really would like to see that go forward, because I think there's a set of creatures that are very unhuman, but have obvious intelligence. I think there's also some really nice work done in cetaceans. So parrots and other birds that have amazing memories and ability to have all these, vast vocalizations. That is a type of intelligence that's different than humans, but is profound and remarkable. Which I guess goes to say that we as stupid humans are learning much more about the intelligence of all the other creatures on the planet, and we're trying to pay attention and address it, and move forward with ways to make sure that we are recognizing that and protecting it. We as stupid humans don't do that very quickly, but I think we are trying to do it. So I think allowing that to go forward and not at the same time tearing down the systems that are trying to do that work is critical. And this is one of the places where we really need to talk about it more than, what we are doing, what we're trying to do better. But this is the challenge, of course, in a setting where we're being punished for doing the work at all. So, I really hope that we as a group can kind of hold up the banner for the importance of the work and trying to do it the best way we can.
Thank you so much, Christy. I think that is like a really important message to end the conversation. I appreciate it. Thank you to the audience members and participants who asked questions. And thank you, Naomi, Christy, and Seth, for sharing your thoughts. This conversation could go on for so much longer. I didn't realize that it was already 1:15. So I appreciate your time and expertise and thoughtful responses. Bye, everyone.
Bye. Thank you.